All right, this is ESCA question 2 for Unit 2. First, we're going to talk about chemoreception of taste and smell. So, first for taste, this is the tongue. Here on top, we have the papillae, and there's these little areas in between the papillae, kind of under it, where we have the taste buds. And the taste buds, they're made up of cells. They're made up of taste bud cell receptors, and there's about 125 taste bud receptor cells in the taste bud. And something that's with these taste bud cells, or the receptor cells, is a lot of times they can get damaged or they kind of just like die. And so these regenerate every 7 to 10 days, and so we're constantly getting new ones. So with these taste bud cell receptors, there's specific ones for specific things that we taste. And the specific things that we taste are sweet, umami, that comes from the Japanese. We have bitter, we have salt, sour, and fatty. Fatty is more unknown, but we did talk about it in class, so I'll talk a little bit about it. And we also have something called capsaicin, and that's a pain receptor that's on the tongue. And just a little side note, I don't know how important this is, but that's what, when we have something spicy, that's what is capturing that. And so it's not like a taste, it's more of a pain that we feel through the capsaicin. And there's a scale that we talked about in class. It's called the Scoville scale. And the maximum is 16 million. That's like the spiciest thing, the max spice. But just for reference, like a jalapeno, I think that's like 5,000 to like 7,500. So not very much. We have a habanero pepper. I think that one was like around, I want to say like 10, 15,000. And then the ghost pepper, which you know is really spicy, I think that one was around like 1 million. So still not even reaching that max of the 16 million. But yeah, back to what we taste. So these things capture certain, when we eat something, there's certain things associated with these tastes that we have, or these flavors, that these taste bud cells capture. So for like sweet, it's like a carbohydrate, or like a sugar, like sucrose or something like that. So a carbohydrate. And we have umami, and that one, I can't remember, but there's bitter, and that one is like calcium. Salt, that's like NaCl. Sour, that's gonna be like acidic stuff. It's like hydrogen cation. Acidic. Fatty, we don't really know, but I think they talked about it's like lipids, but we really don't know a lot about that. But umami, what was that one? I'll have to look it up because I can't remember what it was. I kind of remember it. Glutamate, that's what it is. Glutamate is what sets this one off. Okay, the reason why I clumped these together is because they're specific for one type of cell, and salt and sour are more specific for another type of cell. So we'll do these ones first, sweet, umami, and bitter. So they are going to go to a G protein, the ligand, or it's going to connect there at the receptor point, and the G protein is going to activate phospholipase C, which gives us IP3. Let me explain this a little bit more. This is the cell, the taste bud receptor cell, ECF, ICF. We have a neuron there. still have the SR inside, which has a high concentration of calcium, a high concentration on the outside, low concentration of calcium on the inside. But yeah, one of these three will come in. Again, they're specific. So like sweet, umami, and bitter are going to be specific to this one. One of their chemicals is going to come in. It's going to attach here to the receptor, the G protein, the GPRC I think it is, and it's going to activate phospholipase C, which is going to give us IP3. And IP3, on the SR, there's a specific channel that opens for calcium if it gets IP3. So IP3 is going to come. It's going to allow calcium to come out. So now we have calcium coming out of the SR. But if you remember, with this cell, we have vesicles that are full of neurotransmitters. And with calcium coming in, that's going to allow the snare proteins to connect to the snare proteins on the membrane. And it's going to bring the vesicle to the membrane and allow for the release of the neurotransmitter. And this neurotransmitter is going to connect to a channel on the neuron, which is going to allow a cation to come in, which is going to depolarize the cell and create an action potential. And that's how that message gets sent to the brain. And again, this is specific to sweet, umami, and bitter. So these three right here. These two, we're going to go to this one over here, which is a little bit different. So pretty much for this one, we have a G protein, activates phospholipase, we get IP3, which IP3 activates a channel on the SR, which allows calcium to come out of the SR into ICF, and it allows it to come down here to the vesicles and allow them to attach to the membrane to release the neurotransmitter. Then over here, we have the one for salt and sour, and these ones have a different channel. It's called the ENaC, and that's specifically going to let in either for like the specific one, because they're going to be different for sour and salt, but they're going to let in, one of them is going to let in salt, or Na, sodium. One of them's going to let in sodium, the other one's going to let in, I'm not sure, in class we talked about a video I watched, said something else where with sour, we have the acid or the hydrogen cation, and in the video I was watching it saying the hydrogen will actually block like channels where potassium can leave. So if potassium can't leave, it's going to leave the cell to depolarize. So that was one video said. In class we talked about how the ENaC's either going to let in sodium for a salt specific, and for a sour specific it's going to let in hydrogen cation. But either way, it's going to depolarize the cell. And so for this example, for sodium coming in, it's going to depolarize the cell. It's going to create a Wong, or like a positive electric field. It's going to flow. And on both of them, I'm pretty sure we have the voltage gated calcium channel. That's what we have here. I think it's on both of them. I'm not sure. I think in class I saw it was, but it's not a big player in this one. The big player is the IP3 and the G protein letting the calcium out of the SR. In this one, the Wong's going to allow the voltage gated calcium channel to open. So it's going to allow calcium to come in. And it's kind of the same thing over here like we did. We have the vesicles, have neurotransmitters. The calcium is going to allow those snare proteins to connect and allow the vesicle to be pulled to the membrane. And that's where we're going to release the neurotransmitter, and that's going to activate a channel on the neuron, which is going to let cations in, which is going to depolarize, and we get an action potential along the neuron. So that's how it gets from the foods we eat, chemicals from the foods, to these different channels, to these specific channels for these specific tastes, and it gets sent to the brain. A couple nerves that are important for this, like innervate the tongue. So we have the facial nerve. That's number 7, cranial nerve number 7. That goes to the front 2/3, or the anterior 2/3 of the tongue. So that's going to innervate that. The glossopharyngeal nerve, that's number 9, cranial nerve number 9. That's going to go to the posterior, or the back 1/3 of the tongue. And then the vagus nerve is going to be on the epiglottis. And that's going to innervate that. That's cranial nerve 10. But about the tongue again, the front 2/3 of the tongue is going to have the most amount of taste buds. The posterior 1/3 is going to have less. And then the vagus, or not the vagus, the epiglottis is going to have even less than all of them. But those are what innervates the parts of the tongue and the epiglottis. And that's important because you have somebody come in the video I watched, talked about how if they're not tasting stuff and it's specifically in the front 2/3, you know there's a neurological problem with the facial nerve, or with any of those. You can figure it out depending on the situation. But yeah, that's taste. Now we're going to do smell. So smell, this is my person, this is their nose, this is the nasal cavity. Gotta make sure there's a class starting in here and I don't want to run out of time. I have like 10 more minutes. Okay, so we're gonna rush through this one. Nasal cavity, on the top of the nasal cavity we have the olfactory epithelium. That is also, inside of that, we're gonna have coming from the skull, we're gonna have these, what's the name? It's these nerves anyways. And they're going to be here in the olfactory epithelia. But with something with the olfactory epithelia, it has this mucus on it, this watery mucus. And that's when a smell or like a odorant comes, it's gonna pretty much get attached to the mucus. The mucus is gonna break it up or break it down. And when it gets broken down, the different chemicals are going, or the molecules are going to get picked up by the cilia of this, I can't remember if it's a neuron or a nerve, but it's gonna get picked up right there, and it's gonna send it to the olfactory bulb. And in between that, we have the cribriform plate, and that's where we pretty much have the skull, and then below that we have the nasal cavity. And so there's a plate there, it's a bone, and these nerves go through it. There's like little holes for them to go through. But if someone gets like in a car accident and those break, you can't really restore those. They're gonna lose their sense of smell if they break those and they get in a car accident. Let's say they hit their head really hard and it fractures their skull and it messes with that. If those break, you can't really grow them back or get them back. But a little something I remembered is, when we talked about the taste buds, they regenerate every like 7 to 10 days. These, not the nerves, but these like cells or whatever, they regenerate every, like I think it's like 4 to 8 weeks, maybe like 6 to 8 weeks, but it's a lot longer for smell. But yeah, it goes to the olfactory bulb, and from the olfactory bulb, it goes to the olfactory cortex, and from there it splits off. It can either go to the temporal lobe or the frontal lobe. The frontal lobe connects smells with emotions, which is kind of interesting. The temporal lobe is just when it takes it there and the smell is perceived in this region. Another thing, these don't go through the pathway doesn't go through the hypothalamus like the other senses. But let's take this here, let's blow it up. So what's happening is these chemicals get broken down by the membrane or by the mucus, and they get the cilia hairs pick them up, and when they get picked up, they're brought to a G protein. I think it's GPCR again, and it's gonna pick it up, and it's gonna activate adenyl cyclase, which produces CAMP, and CAMP is gonna go and activate a cation channel and allow an inflow of calcium and sodium, and that's gonna bring them in, which is going to create a Bwong, or a positive electric field. that's going to be sent down the neuron. Talked about this voltage gated chloride channel. I was a little confused on it because it was saying when the BWOM comes, it like allows that to open and it will send chloride out. I didn't really understand that because I thought chloride was supposed to come in because of its driving force and concentration, but I'll have to figure that out and ask about it in tutoring. Yeah, that's smell and taste. I want to get over here to vision. Hopefully I have enough time before people start coming in. But this is the eye. This is the cornea. This is the lens. In front of that we have the pupil. And the important thing here is we want to talk about the lens, because the lens is important for having clear vision. And the lens is kind of sitting here suspensed, and it's connected to these suspensory ligaments in green, which are holding it in place. And then around the lens, and here I drew it so you can see it better. So we have like, this is like a frontal view, looking at it from the front. So here's the lens, and around it on both sides is where that the ciliary muscle is. And that's gonna contract and relax, which is gonna affect these ligaments, which is then gonna affect the lens to either bulge or flatten out when it's getting pulled. So let's talk about that. So, well, the reason why the lens is important is because if we have an object, we're gonna go to right here. If we have an object that we're seeing from really close, it's gonna come, hit the lens, and then the lens is gonna refract that light and bring it back so it hits the back. Sorry, I forgot to mention this. This is the retina. That's where the light hits in the eye, and that's where we are able to change it into signals or action potentials to then see in our brain. But back to this, if we see something close, our lens is going to pick it up, refract it, and then it's going to bend it inward a little bit. And the cornea plays a little bit of a part in bending it, but mostly it's the lens. But if we're really close, you can see it's hitting the lens and it's bending. But if we're really far away, it's still going to bend it. I didn't draw it super well. Kind of messed up, but pretty much the important thing here is when you're really close, it's going to bend at a lot more of an angle. But if you're far away, it's going to, when it hits the lens and it bends it, it's going to bend at a lot less of an angle. And so the important thing here is when we're up close, sorry, one sec. It's important to know if we have something that's concave and light hits it, it's going to bend the light in. So if it's going to hit it, it's going to bend the light in. If we have something that's convex. If we have something that's convex, more bulge, it's going to bend the light in if it's convex. If it's concave, it's going to hit it and it's going to bend the light out. So the reason why that's important, if we're seeing something really close and it's coming at a lot more of an angle, we're going to need to bend that light even more. And so in order to bend the light in even more, we're going to want our lens to be more of a bulge so it bends that light in, because we're getting the light at a sharp angle, so we really want to bend it in good so we can get to the back of the retina. And so that's why, just know if we're seeing something up close, we want it to be bulged so it'll bend the light, because it's coming in at such an angle, we want to bend that light enough to where we can get it to the back of the retina. If we're seeing something from far away, it's coming in at a lot less of an angle, so we don't have to bend it as much. And so we're okay if we have more of a flat or not so convex lens, because we don't need to bend the light as much. Sorry, I might be saying convex and concave, getting them mixed up, but convex bends light in, concave bends light out. So that's why with the muscle and the lens, so it's wrapped around it. If we contract the muscle, it's going to loosen these ligaments because it's coming in. So these ligaments are going to be less tight, which is going to allow the lens to kind of bulge down together. As opposed to if we relax the muscle, it's out more, which is going to pull on these ligaments to pull them tight, which is then going to pull on the lens to pull it tighter to make it flatter. So that's why if we're like seeing something close and we want to bend that light because that angle is so sharp, we want to bend it so it makes it to the back of the retina. We want the lens to be more bulged, and so we're going to relax the muscle, which is then going to make it so the ligaments are loose, which is going to cause the lens to be more bulgy, not so flat. But if we're seeing something from far away, and we don't need to bend the light as much, we're going to want the lens to be more flat, because we don't need to bend it as much, because it's coming in at such a less of an angle. So in this case, we're going to relax the muscle, which is then going to pull on these ligaments to make them tight, which is going to pull on the lens to make it so it's more flat, which is going to make it less bend, make it bend less. And the goal is always to get it right back here on the retina, because that's where the light is perceived at, or it's not perceived, that's where we capture the light pretty much. So that's going to bring us to over here. I have to run through this kind of fast, but I can do another video too. But this is the retina, okay, here at the back. We have these different cells. First we have the, I can't remember, it's like a rod or a cone, and then we have the bipolar cell, and then it goes to this ganglia cell. And the ganglia cell is where it's the neuron that's gonna take it to the brain where it can be perceived for what it is. So on the rod, we have a G protein. I'll just tell you what happens first. So if we have light, how do I want to explain this? Let's just talk through, okay. So if light comes in, we have light, we have this G protein. In the presence of light, it's going to change from instead of the G protein, we have something that's like 11 cis or 11 trans. And pretty much it's this like, I don't even know, but it's like this molecule that is either straight or bent. And at first, it's bent and cis, but when light comes, it breaks one of the bonds and it causes it to be straight, so 11 trans, and it changes the formation of the protein. When it changes the formation of the protein, an alpha subunit is released, and this alpha subunit goes to PDE. PDE, this, if it's activated by the alpha subunit, it will destroy cGMP, cyclic GMP. The reason why cyclic GMP is important, because cyclic GMP is what activates a channel that lets in sodium. And so if cyclic GMP is okay, it's active, it's like not, if it's present, it's gonna open up this sodium channel, which will allow sodium to come in and create a Boang. But if we have PDE activated, it's gonna take out cGMP, which is not gonna allow this channel to open, which is not gonna create a Boang. And if there's no Boang, we're not gonna allow this voltage-gated calcium channel to let in calcium. And if there's no calcium, the vesicles have neurotransmitters will not be able to release neurotransmitter. And there's no neurotransmitter. On the bipolar cell, there's a G protein called mGluR6. And this one, if it's activated, it will block this channel. This channel over here is called TRPM1, and that's a calcium channel. And if this is always open, letting calcium in. If calcium's coming in, it's creating a Boang, and it's gonna allow another voltage-gated calcium to open, which is gonna allow the vesicles to form to the membrane when you release the neurotransmitters to activate AMP to send a signal. But in this case, if let's see here. Yeah, if this is not activated because there's no release of glutamate or no transmitter, this is going to allow this to be open and allow calcium to come in, create a Bwong, let in that, and yeah, go down the cascade. But let's say it's dark. What I first talked about was light, but I'll go through dark. If it's dark, there's not going to be a transformation in GCPR because there's no 11 cis turning into 11 trans. So that is not going to relieve an alpha subunit, which is not going to activate PDE. PDE is not activated, so CGMP is present because it's not being taken out by PDE. So we have CGMP, which is going to activate the sodium channel. Sodium's going to come in, create a Bwong, let calcium in through the voltage gated channel. Calcium's going to allow the vesicles to go to the membrane to release their neurotransmitter. Neurotransmitter is then going to activate mGluR6. mGluR6 is going to block the calcium TRPM1 channel, which won't allow calcium to come in. And if there's no calcium, there's no Bwong. There's no release of glutamate here, which isn't going to activate AMPA, which is not going to send a signal. But this is there's two different types. There's on center, this is the center cell. You also have these side cells. And if it's on center, off surround, then we're not gonna, the bipolar cell isn't gonna have mGluR6. It's gonna have an AMPA. If there's an AMPA, this is now going to be activated by glutamate, whereas before, if there's glutamate, it was going to activate mGluR6, which was then going to block this. But now if we have AMPA here, glutamate's going to come, glutamate's going to activate AMPA, where AMPA is going to bring in cations, which is going to create a Bwong. So it's opposite, you see. So if it's dark, no PDE, is that right? Yeah, there's no PDE, CGMP is activated. If this is activated, it's going to release AMP, or it's going to release neurotransmitter, glutamate, create a Bwong. If it's light, this is going to be activated. Alpha proteins or alpha subunits activated. No this, no release of glutamate, which is going to be no release of glutamate again, which is not going to activate the AMP for the neuron or the ganglia cell. There's no action potential. So pretty much, there can be two types. There's AMPA here. That's off center, on surround. If it's on center, off surround, we have, like we first talked about, the G protein, this CRNP or whatever it's called. That's going to be it for light right now and for the eye. Since no one's coming in, actually, I don't even know if there's a class. So we'll just keep going. Let's actually just keep going talking about this. So we have these, it's called the center cell. We also have these cells on the side. And it's gonna do this, it's gonna have the same process as right here, but instead of going to a bipolar cell, it's gonna have a horizontal cell, which is activated by AMPA. And if it's activated by AMPA, it's gonna release something called GABA. And if GABA is released, it's going to activate this channel right here, which is a sodium chloride channel. And if that's activated, it's either going to let sodium, or sorry, it's going to let potassium, sorry, it's a potassium chloride channel. If it's activated, it's going to let potassium out and chloride in, which is going to, what's it called? It's going to cause the cell to become more negative. It's going to repolarize. And the reason why that's important because it's inhibitory. Because if we're having a Buang here, it's gonna, if we're letting in or like decreasing or lowering the charge, making it more negative, it's gonna cancel out that Buang. We're not gonna have activation of the vesicles with the activation of the, from the activation of the voltage gated calcium channel. So pretty much here, it's gonna go, it's gonna activate. If it's trying to remember. Yeah, pretty much if there's light, it means there's less release of glutamate. If there's no light, it's more release of glutamate. So in like a dark condition, there's more release of glutamate, which is going to activate AMPA, which is going to activate GABA, which is going to activate this potassium chloride channel, which is going to act as an inhibitor for glutamate. There's more to talk about that, but I didn't really draw it yet, so I don't really want to draw it. Go into it, it'll take a long time. But back to the eye, we know that people have farsightedness and nearsightedness. So if someone's nearsighted, it means their eyeball's more long, and if they're farsighted, it means their eyeball's more short. And this is like a normal eyeball. If we have a normal eyeball, it's gonna see something and it's gonna direct it. It's gonna come in, receive the light, it's gonna come in, the lens is gonna get the light, bend it, and bend it to the back of the retina. But in somebody who's short-sighted or nearsighted, they're gonna have a longer eyeball. And so when the lens bends that, you're gonna have a normal lens which is gonna bend it, but since it's long, it's not gonna make it to the back, it's gonna stop short of it. So the vision's gonna be blurry. So in order to make it so it reaches the back, we wanna bend the light out so it can take a different angle. So instead of meeting right here, we wanna bend it out more. So now it reaches back here. So in order to do this, the doctor is gonna give somebody a concave lens, because that's gonna bend the light out. Instead of making it come in so narrow, it's gonna bend it out a little bit, and it's gonna let it reach the back. Someone who's farsighted, they're gonna have the opposite problem where the light's being bent, their eyeball is short, so it's gonna go like to like the back of the eye, not to the back, but instead of meeting at here, it's gonna get cut short. And so in order to correct this, they're gonna give them a lens that's convex because it's gonna bend the light in. And so now we have it bending in more, so it's meeting at the back of the retina. So yeah, that's what I got for taste and smell and vision. And do another quick video for hearing. All right, so this is the hearing part, and up here, got my drawings, and this is an ear. Those sound waves, they travel through the air. Our ear is going to capture it. It's going to funnel it in to our ear canal. It's going to go down, and it's going to hit our eardrum. Our eardrum is very sensitive, so when sound hits it, it's going to cause it to move and to vibrate. Here we have these different bones. Can't remember what they're called, but this is called the stapes. And the first bone is going to move because it's connected to the eardrum. Eardrum's vibrating, it's going to cause this to move. This end's connected to this end, it's going to cause that to move, and finally it's going to hit the stapes, and it's going to cause the stapes to move. And as it's still sending those sound waves, those vibrations are coming through, it's going to come into this part of the ear, and it's going to carry the sound waves, depending on how loud it is, this way further up. This red part is called the basilar membrane. This is like where it's really important to hearing. But if I was to roll it out like this, and just like take it off and roll it out, it would look like this. And on this end down here, it's close to the stapes. That's where the high frequency sound is heard. And up here you can see it's bigger, these bigger lines. This is where low frequency sound is detected. High, 20,000 hertz, low, go down to like 20 hertz, whatever. With age though, you're gonna see this high frequency part begin to deteriorate. So that's why in older people, they often have a harder time hearing high frequency sounds or high pitch noises. Right here's another important part. It's called a round window. It's stretchy. And the reason why that's important is when these sound waves are coming through, it's going to cause movement, but this allows it to, like let's say there's a sound coming through, it's going to allow it to kind of bulb out, which allows for the sound waves to move through this channel. We're going to take this and cut it and slice it in half and look at it this way. This is what we'd be looking at. And we're going to talk about how sound is transformed from sound waves and then through different chemicals, and then how it's finally turned into an electrical signal or an action potential. So here, honestly, you need to study this more. We just learned about it in class yesterday. But this is like the center part. This red part is the bacillar membrane. So you can kind of picture that. Do your best. That's right there. On this side we have the perilymph, and the endolymph is over here. It's not exactly like it, but that's kind of similar to the ICF and the ECF. So you have higher amounts of sodium, higher amounts of potassium. And the reason why we have a high amount of potassium in here is because we have these channels that are, first of all, these are the same, I just didn't want to draw it again. But on this like membrane or channel, this membrane I guess it would probably be, is you have these pumps, pumping sodium out, pumping potassium in. Then also you have these other pump that is letting sodium come in. As it comes in, it's also bringing in sodium, potassium, and chloride. And then you also have more pumps of sodium going out and potassium coming in. I drew that wrong. Pretty much the important thing to know here is we have a bunch of potassium getting pumped in. It gets pumped in here, then it gets pumped again through here. So inside of the endolymph, we have a huge concentration of potassium. And with that, you get a lot of positive charges. And on the other side of this isn't a membrane, this is a tissue, but we still have a separation of charge, and it's of 80 millivolts. But then these calciums are going to come and be attracted, and so we get this separation of charge through this tissue. But on this tissue, we have these little, I can't remember what they're called, these hairs. There's inner hairs and there's outer hairs. But with these inner hairs, or just with these hairs in general, this is what it's kind of going to look like. And here at the top, these hairs, when the vibration comes, they hit against this tectorial membrane and kind of get stuck in there, and they bend a little bit. And why that's important is because when they bend, there's these mechanically gated potassium channels. And let's say it's vibrating through, can't remember the word, resonance. Hit up, it's going to hit the tectorial plate, bend out this way. We have these mechanically gated potassium channels. When that happens, this is going to go that way, which is going to pull. And with this mechanically gated potassium channel, the little tether pretty much connected to this point, or this part. When that bends out, it's going to pull it. And that's going to lift open the lid and allow potassium to come in. And that's happening right here. It's going to hit the tutorial plate, going to open the mechanically gated channels. Potassium has a huge electrical gradient to come out. It's like 120 millivolts. Part of it is due to this, but there's also like another thing that increases it to 140. But it's going to rush through super quick when it hits. So calcium is going to come in. That rush of calcium through that one vibration is going to let in a ton of calcium. And by the way, these vibrations happen like 20,000 times per second, so like crazy quick. Calcium's going to come in super quick. While it's in there, it's only going to be there for a second because there's potassium leak channels. This potassium with these potassium leak channels, it's only going to be there for a quick second, but while it's in there, it's going to depolarize the cell and open up these voltage gated calcium channels, which we've already talked about before with the vesicles and the snare proteins. It connects them to the membrane and allows the vesicles to release neurotransmitter, which then goes to the neuron and to a cation channel, which unlocks it, usually AMPA, and allows for an action potential. But potassium leaves real quick through the leak channel, closes again, and it doesn't release neurotransmitter. But this is happening a ton of times through resonant vibration. Calcium's coming in, depolarizing the cell, letting calcium in, potassium's leaving due to the leak channel, and that's just happening super quick. But this nerve right here that's receiving the neurotransmitter, that's called the vestibular cochlear nerve. And that's running right beneath these hairs. And that's how sound is done. That's how we do it. So yeah, it goes in the ear, vibrations, it's vibrating. These hairs are gonna hit the tutorial plate, gonna open up the mechanically gated potassium channels. And since there's a huge concentration of potassium out here, it's gonna rush in super quick. And as it rushes in, it's gonna quickly cause the cell to depolarize, which is gonna open up the voltage gated calcium channels, which is gonna allow the vesicles to connect to the snare proteins on the membrane and the vesicles, and it's gonna release the neurotransmitters, and it's gonna go to the vestibular cochlear nerve, and it's gonna send the action potential. And it's gonna go to the brain. And that's all I got for you. Like I said, I need to study this a lot more, but this is Essay 2 for Unit 2.